guys for being here. My name is Kate Barnett, and I'm from Montrose High School, and this summer I spent 4 weeks at the George Lab at the University of California, San Diego, studying the neuroscience of addiction. Next, please. So, just a quick preface, my internship is a little bit heavy. It deals with a lot of addiction and euthanasia, so that's just fair warning. So one of the most important studies that the George Lab conducts is called the Genome-Wide Association Study, otherwise known as GWAS. So one of the main goals for GWAS is basically just to find genes associated to addiction, which is a disease. If I remember, I got into an argument with one of the MDs there that addiction was not a disease. So if you take away anything from this presentation, just know that addiction is a disease. And so the end goal is to develop new prevention and treatment strategies because for various individuals, because I would react differently to taking oxy than, say, someone else would. So to do this, the George Lab uses animal models, and those rats over there, they're called heterogeneous stock rats, or HS rats. And we put them in these self-administration chambers. And as you can see, there's like a little wire, and that runs all the way up to a syringe. And in the syringe is either oxycodone or cocaine. Next slide, please. And so there's two levers in these chambers. They kind of pop out like a metal sheet, and so the little rat can like press on it. So usually the rat presses on the drug lever because they get really addicted to it really quickly. And when they press on it in these syringes, a tiny, tiny, tiny bit, I think it's 0.1 milliliters of oxycodone or cocaine gets transmitted from the syringe to the tube down into an IV catheter in their back. That runs a tube all the way up to their jugular vein for direct bloodstream transmission. And as you can see over in the corner, kind of by the paper towels up on the top shelf, there's a whole bunch of wires, and there's a computer monitor in the back that hooks up all these wires to all the self-administration chambers. And they just track how often these rats press, what's their drug intake, things like that. There's also cameras inside the self-administration chambers, which is a new addition to the George Lab study that monitors symptoms. So like, are they irritable? Are they restless? Do they sleep all the time? Things like that. So one of the most important things that the George Lab collects is samples. So over the course of GWAS, when the experiment is running, we collect longitudinal samples. So these are things like feces, blood, urine, to kind of track how much drug they're actually absorbing. And the most important samples are called terminal samples. This is when the experiment is over and we get to euthanize the animals during 3 time points in their drug intake. So either when they're intoxicated or in acute withdrawal or prolonged abstinence. So to do that, we do dissections. I didn't play as much Grey's Anatomy as Avery did, but just a little bit on a small scale. So as you can see over on the far side of the screen, we have our little, Grey's Anatomy setup. And there's like surgical pads, all the stuff you would need to like handle surgery, but on a really small scale. And I mean, basically you just euthanize the animal. I'm not gonna go into too much detail because I'm actually not allowed to talk about it in depth. But there's me, I'm actually pouring a brain into a little mesh sheet and then pouring it into some PBS, which is preservation solution. And so after we take out all of the organs, they go into what's called a biobank. So as you can see, there's like hundreds of tubes. This is only like 1/8 of the room. It's called the Wonder Cave because there's one of my mentors, Lisa Maturin. She covered this room with Wonder Woman posters. So in the Wonder Cave, there's about 1 million tubes, and each tube is designated for a specific organ. So after we put those organs in the tubes, then the tubes go into the freezers, and a freezer farm, which is all the way down there, and these freezers are kept at -80 degrees Celsius, which is really, really, really cold. Like to even open it, you would have to put on a coat and put on gloves so you wouldn't get frostbite. And this is just to preserve all the organs, so like the spleen, even their tail for genetic tracking, and most importantly, the brain. One of the most significant things about the biobank is that the George Lab donates all of these organs to other laboratories who don't have the resources or the funding to conduct this study. So it's kind of like a sharing is caring scientific community. Next slide, please. So I think one of the coolest things I learned was slide mounting, and I think Ayla talked on this just a little bit. So we, quote unquote, pull the brains from the freezers, which just means we take them out, and we put them in a microtome inside a cryostat, which is just basically a big fancy machine that cuts up brains. So then we cryosection the brains, and we put them into PBS, which is the preservation solution. And as you can see, those brain slices they're not cut very well, because as you can see, they're really shredded. So fun fact, if everyone actually would pull a piece of their hair and look at it, one single strand of hair, that is actually 100 microns. So it's pretty small already. But for rat brain slices, we cut them at 40 microns, so you can imagine how frail and how delicate these slices are. And like Ayla mentioned before, we have to use a paintbrush to roll them onto the slide, just so they don't tear. And you pick out certain regions of the brain to mount onto the slide, so like the brainstem, the cerebellum, or the hippocampus, something like that. You mount them onto the slide, and then you let them dry, and then the George Lab keeps them, or they donate some to other laboratories. Some unexpected surprises. You know, in movies, everyone kind of portrays labs as like super ominous and like all work, no play, but that was definitely not the case. I mean, we did work a lot, but I had so much fun at the George Lab. And I also learned that rats are really adorable. Usually the word rat comes with a really negative connotation, but they're actually super cute and super smart. We actually give them like these wooden blocks in their self-administration chambers, and in a couple hours they can chew them into a perfect sphere, like golf ball. It's incredible, yeah. Ready, next slide. For fun, me and Ayla hung out a lot. We visited this one street called Convoy in San Diego. If you guys go to San Diego, I really, really recommend visiting Convoy. It's got a bunch of Oriental restaurants on the street. So we got boba a lot, and I really miss my mom's cooking when I was away from home, so I got Thai food quite a bit. I hiked with my dad because he stayed with me for 2 weeks, and I went to the San Diego Zoo and Safari Park, and I just hung out with my host family. Advice to future you guys: it is okay not to know everything. I mean, like at this age, you're not going to be as smart as like Dr. Olivier George, my mentor, who has like 3 masters and a PhD. Like, you're going there to know it all, or to learn, excuse me. And it's impossible to know it all. And the other thing is, clarity is crucial. Do not be afraid to ask questions, especially when transporting organs. It's really important that you know where they're going, who's getting them, what time you're shipping them out. And a lot of times when researchers wouldn't communicate, it would be really hard and really stressful to find out where the organs were going. And as you can imagine, the amount of resources we expend to, send all these organs and buy all these rats is insane. So it's very important that you ask questions and communicate. I really, really, really want to thank the entire George Lab, especially Olivier and Marcita. And I really want to thank you guys, because this was a life-changing experience, and I would not have been able to do it without you. So thank you, everyone.